Imagine cancer is a company. Now imagine it has a DNA repair department with multiple employees. Each of these employees acts like a genetic repairman to fix broken DNA, which is essential to the survival of cancer. But how can we target DNA repair in cancer? Well, we can model drugs using something called a dominant negative or toxic mutant. Let's take two of these employees, for example. We'll call them Dwight and Jim. Dwight and Jim have the same job, so if one's off sick, another could just fill in. However, Jim is actually a toxic mutant and actively works against Dwight to sabotage him. Similarly, in cancer cells, toxic mutants sabotage DNA repair, ultimately killing the cancer. In my project, I screened a series of mutations in a repair gene called DNA2 and identified two of these toxic mutants. I'm using these mutants to answer two important questions. The first question is, in what types of cancer should we target this gene? Not all DNA repair genes are equally important in all cancers. Some rely on a particular gene to survive. By identifying the types of cancer that are sensitive to these toxic mutants, we can precisely select patients to receive future targeted therapies. The second question is, how should we target this gene? So if I had a pair of molecular glasses that allowed us to zoom really close in on DNA2, we would see something like what I'm showing on the right. And what we see is that it has multiple parts represented in different colors. One part in green is responsible for cutting DNA. Another part in purple is responsible for unwinding DNA. What I found is that mutations that inactivate the cutting function are toxic, whereas if we also take out that unwinding function, we lose this effect. In other words, it's critical that any drugs we develop specifically target the cutting function. To bring my results closer to the clinic, I pass this information to my colleagues who use computer modeling to screen thousands of compounds and identify the ones most likely to inactivate this cutting function. I'm currently testing the 42 most promising drug samples in my lab. In conclusion, my thesis project identified dominant negative mutations in DNA2, which I'm using to model future cancer therapies. So just like employees in the office, cancers have many tools available to repair broken DNA. To take this cancer company down, rather than firing employees one by one, we can use toxic mutants to plant a saboteur and take it down from the inside. Thank you.